Yesterday I forgot to tell you two things because of shortage of time. In fact, to be very frank and honest, it is not that simple to understand in just 50 minutes time. It is not possible for any topic to cover. Eukaryotic transcription is much more complex. Two things I forgot to tell you. Yesterday what I have described is basically for the synthesis of mRNA only. But there are other RNAs also, tRNA and rRNA, and they are produced with the same mechanism, but as I told you yesterday, there is a polycistronic transcription, means at multiple points the transcription occurs, and this tRNA and rRNA are grouped together, clustered genes. They are also transcribed together, and then they are clipped off to make a functional tRNA or rRNA. And on the top you see each nucleotide, whatever we read in the nucleotide nucleic acid chain, it is present in the form of monophosphate, not in the triphosphate. But when DNA synthesis or RNA synthesis is required, DNTP or NTPs are required, ribonucleotide phosphate or deoxyribonucleotide triphosphates are required. So that extra 2 phosphate, that is pyrophosphate, is removed, and that 2 phosphates are cleaved by pyrophosphatase enzyme to release energy and to release free phosphate also. To this point I told you. So basically incoming nucleotide will be basically in the form of triphosphate, but in the phosphodiester bond only single phosphate will be there. Only in the 5' end you can see that is triphosphate. That triphosphate is also subsequently removed. That we will understand in the eukaryotic transcription when there will be capping at the 5' end. During that time, again that pyrophosphate will be removed and additionally 7-methyl guanidine will be added to it. So anyway, yesterday I told you, if you have read it, then the general transcription machinery is the same, and only eukaryotic cell is only one single cell, a limited number of genes of about 42,000 genes, 4,200 genes, and there are base pair, number of base pair is only about 10 lakhs. But in humans we are having, and that 42,187 genes means that much number of protein, that many number of proteins. So single gene usually contributes to single number of protein. Earlier when we were students, we were told that there are 28,000 genes. Now it has been found that 19,000 to 20,000 genes are present. At times some books also write 19,400, something like that. So we are having only this many number of genes, but the number of proteins in a given eukaryotic system ranges from 80,000 to 4 lakhs. So how so many proteins are produced from a limited number of say 1,50,000 genes which are present, honestly speaking, or 19,000 genes. That 1,50,000 is the transcription start site, not genes. So how these are produced? These are basically produced by alternative splicing, post-transcriptional modification and RNA editing, about which I will tell you at the end of this class. So basic principle remaining same. Only one single enzyme was involved yesterday, that was RNA polymerase, and that performs all 5 functions. All 5, it is composed of 5 units along with the sigma factors, and they contribute. So all in one, all factors, say unwinding binding to the DNA site, unwinding, then synthesis of the short chain, extension, then cleavage and release. So, all these things are formed by a single enzyme. In contrast, eukaryotic system, as you know, it is not a single unicellular, it is a multicellular organism, and each organ, even each organ, each cell, each organ, if you say whether it is eye, whether it is liver, the cells are unique and there are multiple cell types of cells are there. The rod cells, cone cells, they also express different types of proteins. Hepatocytes express a different kind of protein than that of what is produced by the pancreatic cells. So, specific tissue specific proteins are there. So, tissue specific transcription occurs to produce that mRNA which are required for that particular cell. So, for that a wide number of transcription factors are there. So, almost 2800. So, meanwhile you write down these differences, salient points. Once you complete it, I will go for the next one. So, there are basic difference between prokaryotes and eukaryotes. These are salient points which I will discuss in this thing. You save the time, meanwhile I draw the picture. You see the site first and foremost, it is in the cytoplasm prokaryotes, eukaryotes it is in the nucleus. And go just write down this thing, then it will be easier to understand. All these things are written in the book. Understood that here basically only one DNA dependent RNA polymerase. This is also DNA dependent RNA polymerase, but this one produces ribosomal RNA and its subunits, that is 20S, 18S and 5.8S. At least remember tRNA, rRNA and mRNA, this part. And this RNA2 is specifically, I am telling you, it is for mRNA, not only mRNA, there are other RNAs are produced also, microRNA, snRNA, long non-coding RNAs. For your brief understanding, I have just written only tRNA, that is also 5S. S means Swift-Berg unit. SVED, Swift-Berg unit. Write down. I will show you one slide. SVED. This is basically when a ribosomal this ribonucleic acid is centrifuged based on its density in a high speed centrifuge. They are sedimented. That is called sedimentation coefficient. Based on that sedimentation coefficient, this has been numbered. So this S stands for Swift-Berg unit as per sedimentation coefficient by high speed centrifugation, even ultra centrifugation also. So as I told you, this is one enzyme, this is 3 enzymes, and promoter region was 10 base pair upstream here, and the second one was 25 base pair upstream. That is 25 base pair, 70 and 90. But 25, 70 and 90 are not fixed, honestly speaking. This can vary a little bit from protein to protein, from mRNA to mRNA, because huge number of mRNAs will be produced. So it is not that only that at that particular specific position there might be 25 to 30, that might be 70 to 80, 75 to 80, that might be 90, 95 like this, and in between also there are residue regions. A sigma subunit was one subunit which consisted of 5 subunits plus sigma factor, sigma unit, subunit. Here there is no such scanning system. This is done by transcription factors, and a huge number of transcription factors are involved. At least I am telling you, there are 2800 DNA binding proteins, out of which 1600 are transcription factors. So it is not a single transcription factor. So that's a huge number of proteins will be produced by the eukaryotic cells, and in the different cell by at least a number of proteins. So accordingly, you can see the 1600 transcription factors will be involved in place of only sigma subunit. The start signal, the TATA box is known as Pribno box. In prokaryote, the same thing is called Goldberg-Hogness box. This is here 25. This is 25 base upstream, 75 base pair upstream, 70, 75 I have written for your understanding. And then termination. As I told you yesterday, it was row dependent or row independent. Here it is done by cleavage polyadenylated specific factor and cleavage stimulating factor. So that is polyadenylated specific factor, cleavage and there is also involvement of the endonuclease. So that basically brings the termination signal, and termination signal is here, this sequence, unlike that polyutil in the prokaryote. Second, post-transcriptional modification, mRNA, which is produced and immediately translated also. There is no post-transcriptional processing or modification. And you will be surprised to know that the synthesis of nucleotide will be at the rate of almost 90 to 100 base pair per second are added in prokaryote. In eukaryote, only 20 to 30 per minute, because it is a highly complex mechanism, and if there is an error in the mRNA, then there will be error in the protein. So high rate of proofreading is there, and that is the reason eukaryotic transcription is very, very slow compared to the prokaryotic transcription. Okay. The action of antibiotics, as I told you, we require to identify specific enzymes or specific biochemical areas where some drugs can be used which will not affect significantly the human being. Actinomycin D was basically used for the bacteria. Now, it was later found to be an anti-cancer agent. So, nowadays actinomycin D is not used for as an antibacterial. It is used for some specific type of cancer which we will read later, and in a very judicious manner. Alpha-Amanitin is by and large used for research purposes to study the activity of the polymerase, RNA polymerase. It is used again in the cancer, it is conjugated with antibody, and that is in the experimental stage also. It is not that much widely used because of its activity of toxic activity on RNA polymerase II, because it inactivates RNA polymerase II, which will basically transcribe mRNA. So, mRNA synthesis will be halted. So, that itself is not used, it is a toxic substance produced from mushroom. Okay. So, 4 main classes, 3 main classes are there, mRNA, tRNA, RNA, plus along with that small RNAs are there. There are number of RNA molecules, small nuclear RNA, microRNA, snRNA, long non-coding RNA. I am not going into the detail. Only thing is that when a RNA is transcribed from the transcription machinery, it is called a nascent mRNA. And nascent mRNA in eukaryotic system contains both coding region and non-coding region, that is called exon and intron. The introns are removed by a mechanism called splicing. So, the exons, basically coding regions, are used, that I will tell at the end. So, as I told you, there are 2,800 proteins in the human genome, but only 1,600 are used as a transcription factors. And all eukaryotic proteins require different types of proteins, and they are, instead of sigma factor, this role is taken by the transcription factor. Now, this process is same again: initiation, elongation and termination. The same structure: formation of the closed complex, then formation of an open complex, replication bubble, synthesis of short stretch of RNA, and then elongation. Then at one point of time, this is the transcription start site, from where at one point of time the termination signal will come, and the termination signal here is AAU, AAA and CA. That is the termination signal. So, why it will terminate basically. So, first and foremost we have to recognize what is the class 1 TSS. Okay. That part needs to be recognized. Additionally, I want to tell you one thing, that in a mature RNA transcript, you will see that this is the transcription start site. This is the transcription start site. Immediately 5' proximal to it, there is one area called 5' untranslated region UTR. Similarly, immediately after the termination site, there will be a region called 3' UTR, untranslated region. This is untranslated, means this is not translated into protein, but this is transcribed. That part is not written in very clearly in your book. So, when this RNA basically binds, this synthetic machinery binds to the proximal, this promoter region, and while the DNA is being unknown, this part is also synthesized simultaneously. So, I am not trying to confuse it, I am just informing that this 5' UTR and this 3' UTR is also a part of the mature RNA transcript. It is not so well described. These perform some amount of regulatory functions and protects also. Okay. So, anything 5' proximal to this transcription structure is called 5' flanking region. Anything beyond this termination point on the right side is called 3' flanking region. This part remember. So don't get confused with the 3' UTR, 5' UTR with the 5' and 3' flanking region. Okay, that part is not that much clear in your book, but for your understanding, at this stage you don't require. So this stage, as I told you, this RNA polymerase will be required, and only recognition of the promoter DNA. How will you recognize? So just like that, yesterday there is a plus minus 25 TATA box is there. Ahead of this, that 75 minus 75 to 80, there is CAAT box, and then still a little ahead, minus 90, the GC box. So basically, the promoter binds here and then goes on scanning. The large structure you will see shortly, not only single molecule, but 20 factors will be attached to it, and it will be a very large complex that will make, because it is very difficult to access a particular portion of the DNA molecule which will be transcribed in a highly complex chromatin structure in the eukaryotic system, particularly human beings. It is not that easy. So that part is very much within the interior of the chromatin, and a huge amount of stretching will be there, has to be there to unwind this process. So that part, this part we recognized first, and then minus 75, and then transcription will start from this transcription start site. And once this is formed, this structure is formed, this RNA polymerase along with the binding factors, that is called basically initiation complex. And then what happens, once this is done, so there will be in that factors, one factor will be endowed with the property of helicase. So that will cause some amount of this type of structure transcription bubble. This will be again formed. This is called an open complex. This I will tell you shortly, and then synthesis will start from here. Promoter will be here. Promoter, once this part is synthesized, at least about 10 nucleotides, this will move to the second one to produce the elongation phase. So again the promoter clearance will be there. Only thing is that you will see that 20 factors will be required to move together, and the entire thing will be released along with only one factor, which is the factor H, which will be bound to the RNA polymerase. Rest all other factors will be released, because the factor H is having three important properties. One is helicase activity, number one, which basically unwinds. Second, it is phosphorylation of the CTD, carboxy terminal domain, which phosphorylation which causes basically activation of the RNA polymerase II, and nucleotide excision repair, because it has to be proofread, and if by chance there is a false incorporation of nucleotide, then that needs to be removed. That nucleotide excision repair is there. So because of that unique property of three functions of the H subunit or factor 2DH, that will move along with the RNA polymerase. Rest all other factors will be removed, because this will go on unwinding. Helicase activity is there. Topoisomerase will also be required here, because severe strain will be there, torsional strain will be there ahead and also behind, because this has to be put back again. So topoisomerase will also be required here. So now I am telling you, once this site is reached, then the cleavage, this area will be reached, and these factors will be attached to that nascent DNA, nascent RNA molecule, and then endonuclease activity will come into play, and that will cleave the nascent mRNA molecule, and it will separate the DNA structure. All the factors will be removed ahead or earlier. RNA polymerase will also be released to make another cycle. That is in short basis the RNA eukaryotic RNA synthetic mechanism. I have told you in a very simple way. Now I will show you in the diagram how complex it is. Once this is formed, it has to undergo post-transcriptional processing. Write down 4 transcriptional processing mechanism. One is 5' capping, 5' 7-methyl guanosine capping, 7-methyl guanosine capping. This is one important factor to protect the 5' end from 5' exonuclease activity, and also it confers stability, and it directs the mRNA molecule to come out from the nucleus into the cytoplasm. So, major part of the transcriptional processing, post-transcriptional modification takes place within the nucleoplasm itself and some part cytoplasm. The number same thing is here, 3' poly-A tail, polyadenylation. Because of the shortage of time, I may not be able to finish it. That's why I am telling you right now. So, 7-methyl guanosine capping is done here. It is triphosphate is there, and then this is basically the first nucleotide. So, triphosphate, first 2 bond phosphate will be removed by a pyrophosphatase enzyme, and then 7-methyl methylation will be done. Guanosine will be added to it, and then methylation will be done by another methyl transferase. Methylation will be done, that will form 7-methyl guanosine, and 3' polyadenylation will be done by the polyadenylate polymerase. Poly-A tail will be formed. So, polyadenylate polymerase will be the enzyme, and once this is formed, suppose once this is formed, so these will be basically having both exon and in between this part there will be introns. This is also intron. Intron region do not code for anything, so that needs to be removed. So, this end has to be cut, this part has to be cut first, this end is also to be cut, then this is to be joined together, and this mechanism is called splicing. Okay, splicing means joining 2 ends together. Of course, after removal, similarly, here it will be added. So, finally, we will get a product of only exons together. Okay. Now, if there is any problem here, if it cannot detect that splicing junction, so abnormal mRNA will be formed. So, abnormal protein. So, this is one of the basic mechanisms of thalassemia and many other diseases. You must understand this thing, and this is easily said there. It actually happens. There are 2 basic mechanisms of intron removal. One is self-mediated, means it does not require basically any ATP, no ATP is required here, no ATP. What happens in this region, there is one specific sequence. Consensus sequence means this is present in everywhere. So, in the middle there is a one signal called A. What happens? This adenine makes a nucleophilic attack here. This is the 3' end of the exon, so it makes a nucleophilic attack and it cuts it. So, this part is cut, this is A, so one structure will be formed like this, loop-like structure, and that is called a lariat structure. Lariat means a loop, just how we keep our animals stacked in through a loop, it is just like that. And then this part will make a nucleophilic attack of here also, this part. This end is there, this 3' hydroxyl is there. So, 3' hydroxyl will make a nucleophilic attack here. So, this part will be removed. So, this is self-mediated. So, basically that lariat structure will be removed and then it will be joined together. That is called self-mediated. And another is where self-mediated genes, it cannot be done. Second mechanism is spliceosome-mediated. Spliceosome-mediated. Say a number of U1, U2, U4, U5, U6 and plus almost plus 60, more than 60 proteins are involved. So, this forms a spliceosome. The mechanism is almost similar here, only thing is that U1 and U2 will bind here, it will form there, then the U4, U5, 6 complex will be there, which is an inactive complex. Once it is bound to this structure, this U4 will be removed, this system will be activated, U6 will replace U1 and that structure will be catalytically active to remove that part, this part. This mechanism is simple, you can read it. In Harper books it is written very well and I have checked it also, your Jambulkar book, you can go through it, it will be easier to understand. Only thing is that you have to remember the sequence, how it is bound. Okay. And then third part and one important part, normally the mRNA which is produced from the nucleus is not edited at all. Now, if you edit something, an alternative structure will be formed. So, RNA editing, see everywhere there is an exception. So, RNA editing is an exception. RNA editing, remember. So far I have told you 3 types, 7' methyl guanosine capping, polyadenylate tail, splicing and last one is RNA editing. RNA editing is that after the mRNA has been formed, now you have editing has been done. Editing has been done to insert a premature stop codon here inside in the sequence of the mRNA. So, actually the full form will not be formed, full structure of the protein will not be formed and a part of the protein will be formed, that is called a truncated protein, not that original protein. So, this is basically one good example is there both in Harper book and your Jambulkar book with apoprotein, ApoB100. Basically what happens, this is a gene which produces ApoB which is having a molecular weight of 100, that is why its name is 100 kDa. Now, at 666 position, there is a stop codon insertion through an RNA editing mechanism by deaminase enzyme. So, here it will stop and it will produce ApoB48. You know ApoB48 is present in the HDL and it is produced in the intestine. So, tissue specific proteins or enzymes are produced through an RNA editing mechanism. Hexokinase is present in the liver, hexokinase is present in pancreas also, but the affinity or Km value is not the same. Km value of hexokinase is not same in the peripheral tissue as that of liver. So, there will be alteration of isoforms. You have seen by this time isoenzymes. So, different forms of the same enzymes are produced having different properties. So, these are all produced by some kind of post-transcriptional modification, either in the form of capping or in the form of this splicing or in the form of RNA editing. Suppose here alternative splicing, how this 28,000 from or 20,000, 19,000 gene, more than a lakh protein is produced. Suppose here what happens, what can happen here? Exon 1, exon 2 and exon 3. Now, instead of cutting here, this exon 1 can join with the exon 3, 1 and 3. It is possible that it has cut here, it has cut here also. It has removed the intervening exon. It is possible. So, 1, 3 joining will be produced altogether a different protein. Similarly, 1, 2, it can be done 1, 2 also. This part is clipped and the fourth one is joined together. Again, 2, 3 can be also there. Just for an understanding I am telling you. So, in multiple combination it can happen. So, each tissue has a specific enzyme, splicing enzyme to splice that side so that the desired protein or the desired enzyme is produced in that particular tissue. See the rod cells, the cells of the skin, even if you look at the skin structure, the structure of the skin everywhere is not the same. Somewhere it is soft, somewhere it is hard, somewhere it is pigmented, some with hair, some without hair, some even with the hair distribution if you see also, it is not universal. So, definitely some amount of proteins and regulations are there. So, that produces a numerable number of basically proteins. Now, I will simply show the pictures, you will just simply go through it and rest you read it from the book. This is the basic mechanism, basic steps remaining same. Only now I will show the pictures very quickly so that I can finish it in another 20 minutes time. So, this part is clear. Now, you see that same thing, transcription, RNA polymerase will be required, that is number one requirement. So, some factors are, transcription factors are required, that is called general transcription factor called GTF, and some specific transcription factor is there which is tissue specific, something for eye, something for muscle, something for liver, something for pancreas, something for stomach, because the functions of the tissue are not same. Some are common, that is why general transcription factor is there. Co-activators or co-regulators. It is not that alone only that sigma factor of prokaryote. Here all these things are required. So, basically it will facilitate promoter binding and the gene expression also. So, activators, co-regulator will basically activate the gene at a given point of time as per need of the cell. Co-regulators will regulate so that excessive production or less production are not there. So, some kind of regulation must be there. This is available in your book. Say RNA 1 thing you note down here, this part, this is not written perhaps. RNAP1 is processed in the nucleolus, RNAP2 in the nucleoplasm, and RNAP3 is also in the nucleoplasm. Everything is happening, all the transcription part is happening within the nucleus, but specifically RNAP1 is within the nucleus and it produces exclusively dedicated for ribosomal RNA, including packaging. full form before it is released. So, RNAP2 in the nucleoplasm, rest of the things whatever will be happening in the nucleus, only the soluble part of the nucleus is nucleoplasm. So, everything will be suspended in the nucleoplasm. So, mRNA, as I told you, including also long non-coding RNA also produced, microRNA, snRNA, and RNAP3 will be for transcription. Now, amanitin, alpha-amanitin, I have told you, it is a toxic substance. It is produced for research purposes mainly. Suppose we want to see which enzyme is getting inhibited. Now, this alpha-amanitin basically, if you have taken 2 test tubes to test whether it is inhibiting RNA polymerase 1, 2 or 3. We want to see. We are doing an experiment of transcription in the test tube. In one test tube, I have not added anything. I have added only the reagents. Another test tube, we have added alpha-amanitin also. Now, you want to see what is the production rate of RNA polymerase. RNA, what is the production? Now, what happens? It is insensitive, means the amount of RNAP produced in the test tube and in the alpha-amanitin test tube, it will have the same thing. It will be insensitive. It will not have any effect. If you look here, RNAP3, there will be some production, but the amount will be less. In RNAP2, there will be a complete stoppage. There will be no production. So, that is basically one part of research purpose. That is what is role of alpha-amanitin, but alpha-amanitin of late has been used since this completely inhibits RNA polymerase II. So, in cancer cell, when there is excessive synthesis of cells, the cells have to be stopped either at the replication, transcription or translation stage by any means. So, this is one mechanism of stopping RNAP2, means mRNA will not be produced, the cell will die. So, that can be tagged with an antibody, so that it will be targeted therapy. Only cancer cells will be affected, not the normal cells. Otherwise, if the normal cells are affected, then again the synthesis will stop in all the cells also. So, in cancer therapy, the major requirement is to target the cancer cells. There are many rapidly proliferating cells in our body, like bone marrow, like gonads, like intestine, like skin, but the skins are rapidly proliferating. So, whenever any anti-cancer drug is given, usually bone marrow is depressed because some amount of hematopoietic cell production will be depressed. There will be sterility also, because gonadal cells will not be able to produce germ cells, even gastrointestinal bleeding and bleeding from the skin, because all these cells are highly proliferating cells. So, there is a possibility, if you give the anti-cancer drug at a very high dose, there is a possibility of bone marrow depression. That is why regularly the WBC count, RBC count, hemoglobin levels are checked while the patient is on chemotherapy. And we have to check the coagulation profile completely before we start any anti-chemotherapy, before any cancer chemotherapy, because there is a chance of bleeding. Okay. So, this RNA polymerase II is basically, this one I told you, only thing is that a human ribosome is consisting of only 80S ribosome. It is having 60S and 40S subunit. I will tell you the importance in this protein class. As bacterial ribosome is having 70S, but it is having 2 units, that is 50 and 30S subunits. As safe drug, I have already explained. So, RNA polymerase, you have seen that is having only 5 subunits. Now, you see look at the 42 proteins are involved. So, RNAP1 is having 13 proteins. RNAP2, which is of prime concern for mRNA synthesis, is having 12 subunits of proteins. You see each protein is having some amount of functions. So, RNAP3, which produces tRNA, number is having 17. So, 42 proteins are involved as a transcription factor, the integral part of the RNA polymerase basically, not transcription factor. So, RNA polymerase II is a multi-protein complex. The structure is very critical. As I told you, that one part of the H function is phosphorylation. This is a heptapeptide unit, that is called, this is 52 heptapeptide or heptide are there. So, this RNA polymerase II is endowed with heptapeptide and phosphorylation also occurs here. So this is the heptapeptide sequence, and the activation is by phosphorylation of the serine and threonine of the carboxyl terminal domain, and that makes the enzyme active. And helicase H unit, factor 2, you will read in the factor 2 for RNA polymerase mRNA 2, factor 2 means it is related to RNA polymerase 2. When it is a factor 1, that means it is related to RNA polymerase 1, and if it is a factor 3, means it is related to RNA polymerase 3. The namings are for that. So RNA polymerase 2 means basically it is related to that second polymerase. So this processing will, this first step I have told you. Now this is serine and threonine will be involved in the elongation process. So it is a complex transcriptional machinery, as I told you earlier. So 12 subunits of RNA polymerase require 6 transcription factors. That is this TATA box is basically called TATA binding protein. The TBP is an universal transcription factor because TATA box will be there in all mRNAs. And in TATA box, in the first stage, the TATA box here, the TBP will bind here. TBP will combine with factor 2D along with 14 associated transcription associated factors. So that will form first TATA binding protein will be there, to which factor 2D will be attached, factor 2D will be attached, and it will cause a bending, sharp bending. So it will form a bending leg structure. That transcription bubble, what I have said you, it will cause a bending. Now then factor 2D is there, then subsequently factor A, factor B, this is 2A, 2D is already there, A, B, then factor C is not there, D is already there, then E is not here, F is there. F is endowed with RNA polymerase 2. F, then E, and lastly H. This H is unique. H is having 3 functions, as I told you. So once this bending has taken place, then there will be unwinding and formation of that open complex. So you see here this TATA binding protein is there. TATA binding protein will form, this will be attached to it, TATA binding protein along with transcription associated factor and factor 2D. So there will be almost 15 numbers of transcription factors are now already attached to it. Now subsequently A will be attached, then B. Remember the sequence only. First to D, then to A, then to B, each is having a different function, and then to E. To F will be there, F will basically associated with RNA polymerase 2, and then E will basically stimulate the formation of ATPase, and helicase activity is there in the A, that is H. This H is endowed with the 3 important functions, as I told you. It unwinds the DNA polymerase, so it is having within helicase property, it is having a kinase activity, that is phosphorylation of RNA2 within the CTD, that will cause activation of the RNA polymerase 2, and it recruits nucleotide excision protein, means it has, if there is by chance if there is a false insertion, this has to function like this. So ultimately what happens basically, so at this stage, if there is a false insertion, this is I am telling you later. So these are the counter basal components, co-regulators and activators. Now role of activators and repressors. So activators will basically bind with this structure, and it will bind to the promoter region to cause activation, and repressor, if it is function, so it will bind to the another promoter region, it will cause repression. Suppose this when the activation will be there, so this activator will form at this site, and this way transcription will be increased, and at the same time repressor molecule will bound to this thing, so that there will be no repression. So just reverse will occur when there will be repression of the gene. So mediators are basically this is enhancer region, which is quite away from that promoter binding region. This is attached to it activators. These activators can bind to this thing, this promoter region, through a mediator. So that will enhance, this is called an enhancosome. It will increase the synthesis of the mRNA. How polymerase will know what to start, that is what I have told you, that we have to see that at where is the TATA box, where is the GC box, where is the CAAT box. So how often it will be transcripted as per the need of the cells, the signal specific factor frequency of the signals are there. Now eukaryotic region, promoter proximal region, I have told you this is this region, this part is the activator repressor, this is promoter proximal region is also there, this part. So enhancer region is there, repressor region is there. So promoter has to be accessible, one thing. It is a complex structure, as I told you, nucleotide, it is a very complex, chromatin is a very complex structure. So we have to get an access to that area. So promoter accessibility to the particular area of the nucleotide, that sequence of the DNA, the transcription initiation will take place. This part I have discussed, formation of pre-initiation complex, then initiation of RNA synthesis, and the promoter area will be cleared also. So transcription unit I have showed yesterday. So key features are that basically the entire unit which is involved in the transcription is called a transcription unit. So everywhere this, it is not that GC will be here, GC can be there also. This is a thymidine kinase gene also, you can see that GC region is here also. So it is not that even, this is general idea. So that bending is the occur pre-initiation complex. There are functions of the different A, B, C, D. This is related to RNA polymerase II. That's why its name is factor 2A, 2B, 2D. And helicase activity, I am telling you repeatedly that helicase will be bound to the only with the RNA polymerase for elongation to occur. So bending, initiation of the synthesis, this is formation of bending of the DNA by the TATA binding protein, then attachment, serial attachment of all the factors, then pre-initiation complex, then unwinding of the DNA open complex, initiation of the RNA synthesis, then the promoter area will be cleared also. So pre-initiation complex will be that, it is this much complex schematically, in reality it is much more complex. Now then elongation will start, this CTD is phosphorylated, this is remember, CTD requires phosphorylation for the RNA polymerase II to be active. So elongation will take place during elongation phase. Why there is so much of delay of 10 to 20 to 30 nucleotides per second? The proofreading is one important part. When the proofreading is there, here one thing again I forgot to tell you that the RNA synthesis, when it will start, it will not be in the very first stage. There will be several abortive attempts, because this is almost a 20 complex compound. The RNA DNA hybrid will be an unstable one. You have to check it whether promoter, that RNA polymerase II can be cleared from that area completely or not, or whether the DNA RNA hybrid is formed and is destroyed. So some kind of checking mechanism is there. So some attempts will be basically an abort, that will be aborted, that is called abortive pre-initial part of this thing in this transcription complex. Once it is found that, well, a stable DNA RNA hybrid has been formed, then only the other factors will be dissociated, because 20 factors is attached to it, the DNA hybrid complex is a small amount. So during when it is getting separated, the high chance that the DNA and RNA hybrid will be destroyed, as for the initial part of the RNA polymerase molecule will be destroyed. So that part is important. Similarly here, when during elongation phase, when there is an incorporation of false, there will be pause, immediate pause, and it will try to rectify it. So it will remove the nucleotide it will excise and then it will replace again. So there will be pause, there will be backtracking again. If there is a checking point during checking time, so there will be pausing, there will be pausing. Well, how much mRNA has been transcribed? Is it adequate for the need? Then it will be, there will be pause for some time, or backtracking if there is any false incorporation. So that is the reason why elongation takes so much of longer time, because mRNA has to be produced in a perfect form. So termination will occur, as I told you, this cleavage and exonuclease activity and endonuclease activity will be there. So this will be recognized, this AAU will be recognized by the CPSF, and the CA will be recognized by the cleavage stimulating factor. So the RNA molecule will be released, and termination will occur through a polyadenylation phase. So polyadenylation signal will be there and it will be stopped basically. Okay, so this will be regulated just like prokaryotic transcription. Some kind of regulation must be there, and this is altogether a different chapter. So post-transcriptional modification, I have told you earlier that you remember this 5-part 5' methyl 7-methyl guanosine capping, then poly A tail addition to protect 5' exonuclease activity, to protect from 3' exonuclease activity, to confer stability, and to allow the mature mRNA or almost mature mRNA to come out from the nucleus into the cytoplasm to function properly, because ultimately basically tRNA, RNA and mRNA and other RNAs will function mainly in the cytoplasm itself. So it has to come out from that thing. So basically everything is, all the post-transcriptional modifications take place in the nucleus itself, and if it is not done, it is not complete, rest the functions are done by at the cytoplasm level only. Okay. So this basically there are endonuclease cleavage, poly A tailing, capping of everything I have told you. This exon neutron, exon removal I have told you also. So this part also I have described 5' untranslated region and 5' flanking region, 3' flanking region. So this is basically protein coding region, and I told you that 5' UTR and 3' UTR is also a part of total thing, but it is not described classically during the transcription phase. So this will come out, this is in the nucleus, this will come out through the nuclear pore into the cytoplasm. So a good number of this spliceosome, I told you that U is there because of the high amount of uridine in that RNA molecule. That's why it is named as U1, U2, U6, U3, U4, U4, but U3 is not required, is not involved in the spliceosome formation. So these are present in the nucleoplasm and nucleolus. Nucleus, you see that U3 is present in the nucleus, and this part is present in the nucleus and cytoplasm. So snRNA formation, basically uracil region, that is uridine, that's why its name is U. U name is there. So there will be more than 60 small nuclear ribonucleoprotein. That's why it is called snRNP, small nuclear ribonucleoprotein for spliceosome. Okay. So this is basically how it is formed. That lariat structure I have told you. Alternate splicing I told you. Again, there are 3 exons example I have given, 1, 2 or 1, 3 or 2, 3. If there is a mutation, there will be again disease just like thalassemia I have told you. Alternative splicing, originally the cognate DNA, it will be basically copied as mRNA, but RNA editing if it is done post-transcriptionally. Then see apolipoprotein B, this is a molecular weight of 100A, so it will produce a B100. Now B48, there are premature stop codon at 666 position. So this is CA will be basically replaced by UAA through a deamination, cytidine deaminase basically is there. So it will form a stop codon, it will cause a truncated protein. Any protein which is not present in its original form is called native form. The desired form is called as truncated protein. Okay. With this I add, I think you read it, hopefully you will understand, and for your own examination purposes I will tell you, we make a chart between prokaryotic transcription, the steps, write down the differences, particularly the inhibitors, because the inhibitors of transcription, replication, transcription and translation might come and likely to come as a short note or an MCQ question. Where it is inhibited, that part you must make a note, and just before examination you just read that thing, otherwise it will be very difficult to remember. You cannot remember it. If once reading it is not possible. You reserve that page at least 1 hour before the examination, so that you can really write it. So it is very difficult, I am telling you, to remember all these things. You cannot. You read, make a note in the way you like, and you will be able to write something during the examination. Okay.